After talking about how the impulse is being generated, a little bit about the structure of the neuron and how these impulses are moved from one point to another. Now we'll move to the next topic, that is the structure of synapses. So axons and all of its branches may form junctions with the dendrites of other neurons, with muscle cells or with gland cells called the synapse. So this is basically a connection or a juncture point where two neurons are attached, or neuron is attached to muscle or other gland cells in order to transfer the impulse. So this is an electron micrograph. So this is the post-synaptic part. So here you can see this is the axon terminal. So here in the axonal terminal, and this is what we call as the post-synaptic cell. So this is skeletal muscles, and this is the space which is between the axon and the skeletal muscle cell. So this is known as the synaptic cleft, and in this axon terminal, there are a lot of vesicles present, which are known as the synaptic vesicles. And of course, this is what we call as the mitochondria. So now let's have a look at the pictorial depiction of what we saw as the electron micrograph. So here, this is the muscle fiber. So this muscle fiber has a lot of protein receptors. These receptors, they are specifically designed for identifying the or for binding of the neurotransmitter acetylcholine. So here, this is what we saw in the previous slide, the axon. This axon has a lot of synaptic vesicles. In those synaptic vesicles, there are small amounts of acetylcholine, or you can say there are small depots of acetylcholine. And this is what we see is the synaptic cleft, which is the space between the axon and the muscle. And this is, okay, I'm sorry. So this is the covering of the muscle cell, which is called as the sarcolemma. It is something similar to what we call as plasma membrane, and also it has mitochondria and other organelles. So now, when impulse reaches the axon, so that impulse triggers or facilitates the uptake of calcium. Now, this calcium uptake further leads to a trigger of events that facilitate or that allows the fusion of the synaptic vesicles with the axon terminal. So as the synaptic vesicles fuse with the axon terminal, the acetylcholine is being released. Now this acetylcholine travels via the synaptic cleft and reaches to their specific receptors. And after binding to the receptors, they lead to the opening of the channels in the postsynaptic membrane. So this is how the neurotransmitter acetylcholine is released, and the impulse is transferred from the axon to the muscle cell. So acetylcholine, it was the first neurotransmitter which was discovered, and this crosses the synapse between the motor neuron and the muscle fiber. Now this synapse between the motor neuron and the muscle is known as the neuromuscular junction. So this is a typical micrograph where you can see one of the axon is connected to a couple of neuromuscular junction. So there is branching in the axon here at this particular point, and it is attached to 3 of the neuromuscular junctions. Now here you can see a little bit of detail of what we see in the previous slide. So in the synaptic cleft, we have a lot of acetylcholine molecules. Now this acetylcholine molecules, they'll bind to the receptor which is present on the muscle cell at the neuromuscular junction. So you can see this is a specific site where the acetylcholine is going to bind. And initially, before the binding of acetylcholine, this particular receptor protein, which is also having a ion channel, is closed. So here what we see as the closed structure. Now, when this acetylcholine binds to the specific point, the point of binding of the acetylcholine, this leads to the opening of the channel. Now, because of the opening of the channel, now as I told you earlier, there will be uptake of sodium. So 3 molecules of sodium will be taken in and 2 molecules of potassium will be moving out. So that movement of sodium will lead to the change in the overall charge inside the membrane. So a positive charge will be generated. And now this from negative to positive charge will lead to the movement of the impulse in the muscle. Okay, so we can have different neurotransmitters and they can have different kind of effect. So excitatory neurotransmitter promotes depolarization. So depolarization means generation of the charge or generation of the impulse that will take place when there is a impulse or there is a neurotransmitter which is there to promote the excitation. So we call it the excitatory post-synaptic potential. So let's have a look at this particular diagram here. So this is what we see as the axon terminal, and this is the muscle or the other axon terminal. So here we may have the neurotransmitter which is responsible for generating a excitatory post-synaptic potential. So now this vesicle fuses with the membrane of the axon terminal. They are released into the synaptic cleft and they bind to the specific receptors. So now this binding would lead to the opening of the gate, and that will lead to the uptake of the sodium inside the channel or inside the terminal of the postsynaptic, and that will lead to the increase in the membrane potential. So excitatory postsynaptic potential will change. Our impulse will be generated or the change in the electric potential can be detected. On the contrary, there could be some inhibitory neurotransmitter and that would bind to its specific receptor which are present at the another postsynaptic terminal. So now this binding to another specific receptor will lead to the opening of a gate that will promote the uptake of chloride ions. So now these chloride ions, they will generate a negative charge inside the membrane. So that generation of negative charge will be called as the hyperpolarization. And this hyperpolarization can be detected via electrical events. So there is a decrease from the resting membrane potential, that is minus 70 millivolt, and that can easily be detected and identified. It is because of the uptake of the chloride ions. So acetylcholine stimulates muscle contraction. So if the acetylcholine is doing that, so there should be some way that this acetylcholine should be removed so as the stimulus or the muscle contraction which has been generated due to the binding of acetylcholine is removed. So if that stimulus will be removed, then only the muscle will come to the relaxing stage. So in case of acetylcholine, there is an enzyme which is named as acetylcholine esterase. So this enzyme, acetylcholine esterase, at the postsynaptic membrane. So this deactivates the acetylcholine into smaller fragments, and that smaller fragments are inactive. And as soon as this acetylcholine acts on the acetylcholine, this acetylcholine is degraded and the impulse which was generated due to the binding of acetylcholine is also broken, or that impulse is also degraded, and immediately the muscles comes to the rest. Now there are other molecules also like glutamate, glycine and GABA. So Glutamate is the major excitatory neurotransmitter in the vertebrate of central nervous system. They produce excitatory postsynaptic potential and they have a stimulating action at the postsynaptic neurons. However, excessive stimulation by glutamate may cause neurodegeneration. So one of the example is the Huntington's chorea. So another molecules like glycine and GABA. So GABA is an acronym for gamma aminobutyric acid. So these have inhibitory impulses or they are inhibitory neurotransmitters. So they cause the opening of the channels which regulate the uptake of chlorine. So when the chlorine channels they are open, there will be uptake of chlorine and the resting membrane potential from -70 will further drop down to -85 millivolt. And this hyperpolarization is called inhibitory postsynaptic potential. Now there are other molecules also which we call as biogenic amines. So this biogenic amines include hormone epinephrine or adrenaline, and also they have neurotransmitters like dopamine, norepinephrine, and serotonin. So epinephrine, norepinephrine, and dopamine, they are derived from amino acid tyrosine, and they are together counted under the subcategory of catecholamines. Serotonin is a biogenic amine. It is derived from amino acid tryptophan. Dopamine is a very important neurotransmitter in the brain and it controls body movements and other functions as well. Now, the degeneration of dopamine releasing neurons produces Parkinson's disease, which is characterized by muscle tremors. You might have seen some people, they cannot control, especially the old people, they cannot control the movement of their hands or sometimes neck. It continuously keeps on vibrating. So that is the symptom of Parkinson's disease. Usually it is treated by giving L-dopa. So that is a precursor of dopamine. Since there is decrease in the amount of dopamine and amount of dopamine is given from outside. Now there could be possibility where excess activity of dopamine releasing neurons is also there. And that particular situation is called schizophrenia. So usually these patients which are suffering from schizophrenia, they are given a drug that could block the production of dopamine. So, or just inhibiting the production of dopamine is kind of considered as a treatment of schizophrenia. So, norepinephrine is used by autonomic nervous system in the brain. So, it acts as a neurotransmitter, complements the action of hormone epinephrine, which is secreted by the adrenal gland. Serotonin is another neurotransmitter, and this is involved in the regulation of sleep and also in other emotional states. So if there is insufficient amount of serotonin, it may lead to different kind of clinical symptoms. So one of the major is the depression. So this is what we see is that here you can see. So this axon is releasing serotonin. So now this serotonin binds to a specific receptor. So if there are less amount of serotonin release, it will lead to depression. So there is drugs like Prozac, which are known as antidepressant drugs. So these antidepressant drugs, they are given so that even the small amount of serotonin which is released by the axons are not reabsorbed. They are continuously available to bind to the specific receptors at the terminal of the postsynaptic neuron. So this binding of the serotonin at the receptor will keep on giving the action, or if the serotonin amount could be increased, that would also lead to the enhancement or removal of the depression. So there could be two probable here: either the serotonin release increase, or it is made available to the receptors by inhibiting the reabsorption. So this drug Prozac, it inhibits the reabsorption. So there are other neurotransmitters as well. So exons, they also release a variety of polypeptides, which are called as neuropeptides. So at the synapse, they may have different kind of roles, and they may have long-term effects also on the post-synaptic neurons. So they are usually modulating or affecting the action at the neurons. So they are also known as neuromodulators. So one of the important neuropeptide is substance P. So this is released at the synapse in the central nervous system by the sensory neurons, and it is activated by painful stimuli. So the intensity with which pain is perceived partially depends on the effect of neuropeptides called enkephalins and endorphins. So enkephalins, they are released by axons which are descending from the brain and inhibit the passage of pain information to the brain. So enkephalins, they are kind of playing a prominent role so that the pain is not perceived by the brain. So endorphins are released by neurons in the brain stem, and also they block the perception of pain. So if there is sufficient amount of release of endorphins and enkephalins, the person will not feel pain. Now there are drugs which are derived from opium, like morphine and heroin. So they have a structural similarity with the enkephalins and endorphins. So because of this structure similarity, they bind to the specific receptors where endorphins and enkephalins are supposed to bind, and exert the similar effect as they are doing. So they are having a pain relieving effect. So usually if somebody has an accident or they have some kind of wound, then these opium derivatives, they are usually given to mask the pain which is being generated because of that injury or wound. So nitric oxide, it is the first gas known to act as a regulating molecule in the body. Now, since this is a gas, it cannot be stored in the body. So it is usually produced when it is required, and it is produced from amino acid arginine. So nitric oxide diffuses out of the presynaptic axon into the neighboring cells by simply passing through the lipid portion of the cell membranes. So in the peripheral nervous system, nitric oxide is released by some neurons that innervate the gastrointestinal tract, penis, respiratory passage, and the cerebral blood vessel. So in autonomic nervous system, nitric oxide has a smooth muscle relaxation role. If you remember from the previous lecture of the endocrine glands, there also I told that nitric oxide is responsible for the vasodilation. So when it is leading to vasodilation, there is improved movement of blood. So if let's say we talk about the GI tract, so there vasodilation leads to more flow of blood when the digestion process is taking place. So that would lead to absorption of more and more nutrients. So nitric oxide is also released as a neurotransmitter in the brain, and it has been associated with the process of learning and memory. So one can imagine that nitric oxide has various roles. So it is quite different from nitrous oxide, which is N2O, that is called as the laughing gas. So that laughing gas is usually used by some dentists, or maybe to disperse the crowds. They are used by police personals. Okay, so now we come to the synaptic integration. So it is quite possible that at a specific junction, there may be some axons which are excitatory, let's say in this case, blue ones, and some which are having a inhibitory response. So now, depending on how many excitatory or how many inhibitory axons are there, so the influence of excitatory and the inhibitory, it can be summed up. So the total can be done, and the overall inhibition or excitation of the postsynaptic neuron will depend on how much is the excitatory or how much is the inhibitory. If the total of that inhibitory and the excitatory comes out to be sufficiently enough to depolarize, then the action potential will be produced. If that total is not sufficient enough, there will be no depolarization. So this is electron micrograph, where you can see there are a lot of axons which are binding to the postsynaptic neuron. Okay, so let's move to another topic, which is called neurotransmitters and drug addiction. So when a cell is exposed to a stimulus that produces a chemically mediated signal for a prolonged period, it tends to lose its ability to respond to the stimulus with its original intensity. So let's say, for instance, if let's say somebody is exposed to, let's say, or somebody is taking a very spicy food. So if he's taking that spicy food for longer duration, he will not be able to enjoy a food which is having a medium or low amount of spices. Why? Because the receptors which are in the tongue, they have that kind of sensation that in order for them to activate, they need a high level of spices. Similarly, it's the case of receptors here. So if one receptor is stimulated by a large amount of chemical mediator, it will not be able to respond to a specific receptor, to a specific stimulus given by the original molecule. Okay, so let's see an example of a drug. So that example could be a drug cocaine. So this drug cocaine, it is usually acting on a transporter protein, which is responsible for the uptake of the dopamine. So now, as you know, that dopamine, it is kind of a neurotransmitter which is associated with the pleasure. So the pleasure pathway, or also known as the limbic pathway, is affected by the cocaine. So in normal instances, this dopamine is released, it binds to the postsynaptic neuron and exerts the feeling of pleasure. Now, after doing its job, it is being transported by the transport proteins and taken up by the axon. However, in case of when cocaine is being taken up, so this cocaine binds to the transporter proteins. So now the transport proteins, they are unable to bind to dopamine, and dopamine exerts its effect on the postsynaptic neuron for longer duration. So because of the uptake of this cocaine, the person has a feeling of pleasure. Why? Because the dopamine is being not absorbed or not reabsorbed by the axon terminal, and is available to bind to the receptor in the postsynaptic neuron, and the feeling of pleasure is prolonged. Okay, so let's see this particular slide here. So here, this is a normal instance where this neurotransmitter is being released. It is transported by a molecule and taken up by the axon. So, however, in another instance where a drug molecule is taken, let's say for example, we talk about the cocaine. So, this cocaine will bind to the transporter protein. So, this binding will prevent the reabsorption of, let's say, dopamine here, and that will lead to the increase in the pleasure time. However, now because of this, the prolonged excitation or the prolonged availability of the dopamine, the neurons will respond back. So what will they do? They will reduce the number of receptors. You can see here, there are a couple of receptors present. So their number of receptors will decrease. Now, at that particular instance where the lot of dopamine was available, even the small number of receptors will be good enough. Now imagine in the instance where the cocaine is not being taken, or a person is not taking that particular drug. Now, if he is not taking that particular drug, now the transporter molecules, they are freely available. So these transport molecules, they'll bind to the dopamine and it will be reabsorbed by the axon. Only a small amount of dopamine will be available. Now there are two instances. The amount of dopamine is released is less and the amount of receptors are even less. So the person will have a problem of addiction. So in order to have pleasure or antidepressant effect, the person will need the drug again, or he'll have something which called as withdrawal symptoms. Okay, so after talking about, let's talk about a little bit about the vertebrate brain. So the cnidarians, here you can see the hydra. So the cnidarians, they have the simplest of the nervous system. They have a network or a web of nerves which call as the nerve net. So if we move a little bit further to higher organisms, so the simplest animals that they could have the nervous system developed, one is the platyhelminths or the flat worms. And if we move a little bit higher, let's say we talk about the arthropods. So it is the arthropods where there is a complex coordinated responses there. So usually in the arthropods, there is a front nerve cord. So that nerve cord is divided into smaller nerves and is passing to the lower part of the body. So it is the terminal part or the anterior part which is controlling the movement or which is controlling the nervous system which is being innervated into the different parts of the body. So it is in the humans which is the nervous system is highly developed, where the anterior part of the brain has the different components which control the body parts or innervated to the different parts of the body. The basic organization of the vertebrate can be seen in the brain of primitive fishes. So in fishes, the brain is divided into three regions. So what are those regions? These are the hind brain. So you can see from here, this part, the hind brain is the largest portion of the brain. So that hind brain constitutes of medulla oblongata, pons, and the cerebellum. So later part, that is the mid brain, is devoted primarily to the visual information. So here we could have the different part, that is the major is the optic tacta. The forebrain is mainly concerned with olfaction. So here you can see the forebrain part. So here the main is the olfactory bulb, which is part of the cerebrum. So this is kind of a basic organization of the brain in the primitive fishes. So probably we'll stop here and we'll continue it in the next lecture. So thank you very much for your attention.